welcome you all to my lecture series on lichens, part 3. This topic, lichens, comes under plant diversity 2 with the course code 23U2BCCT2. In today's lecture, we will be dealing with the structure and life cycle of Usnea. Before getting into the details of Usnea, structure and life cycle, let me show you some of the images that we must understand to know the habitat and the morphology of Usnea with these images. Usnea serratina, you can see there is a bark and then you have a hanging Usnea thallus. And here in this image, where it has been taken in Baikal Nature Reserve, Russia, pendulous Usnea lichens are seen in the dense forest or the jungle. In here, Usnea dasypaga, here also in the deep jungle you can see there is pendulous hanging Usnea thalli. And it's another species, Usnea cavernosa. This is again an image taken in a jungle or a forest where these lichens are present in abundance. So as far as the morphology is concerned, this Usnea thallus, it belongs to the fruticose lichens that are commonly found hanging from the branches of the trees which are present in moist, clean forests. It is popularly known as old man's beard or beard lichen due to its characteristic hair-like pendulous growth which we have seen in the previous slide, and it is attached to the trunks of the trees by rhizoid-like outgrowths which are referred as rhizines. The plant grows erect and pendants with the main branch which are bearing so many lateral branches, and the color of the thallus it varies from grey, green or greenish yellow. There are several conspicuous bristles or fibrils you can see on the thallus, and that covers the thallus entirely. The lateral branches terminally bear a large plate-like structure referred as apothecium, plural apothecia. Their margins are fringed with bristle-like outgrowths that may be simple or sometimes they are branched. It serves as an excellent model organism to understand the biology of fruticose lichens. So now we get into the details of the internal structure of Usnea thallus. Here you can see the hanging pendulous Usnea thallus, and you can take transverse section or cross section in any of these regions, and you will come across a beautiful cross section where we can start from the central cord, which is otherwise referred as chondroid axis. This is the most distinctive feature of Usnea. This is a very strong, elastic, at the same time cord-like structure that runs through the center of each branch. It is composed of tightly packed, thick-walled fungal cells that provide remarkable tensile strength, at the same time flexibility. This axis prevents the lichen from snapping easily in the wind, much like a wire within a cable. And now you have the second region which we call as medulla. Surrounding this central core is a region medulla. This layer consists of loosely interwoven fungal hyphae with large air spaces between them. Its primary functions are to act as a sponge, absorbing and holding water. Not just that, to facilitate the exchange of gases. And now you have the algae layer, or we can call also as gonidial layer. Encasing the medulla is a thin layer where you can see the symbiotic partner resides. This layer contains the photobionts, usually a green alga or green algae, entangled in a dense network of fungal hyphae. This is the primary site for photosynthesis because it is an alga, or you can see algae, so they are primarily involved in photosynthesis. And the final or the outermost layer, which we refer it as cortex, the outermost layer. It is made of densely packed gelatinized fungal hyphae that form a continuous protective as well as waterproof coat. This layer protects the inner algal layer from excessive light and reduces water loss also. So a simple analogy I can give you, think of Usnea strand as an electrical cable. The central axis is the copper wire, which is meant for strength. The medulla is an insulation, which is meant for storage and cushioning. The third, the algal layer, which is a thin vital film wrapped around it, which is a productive unit. And finally, the cortex is the outer plastic sheath for protection. So here are the English terminologies and the Tamil terminologies. They are along with their main functions. And now we get into the details of the life cycle of Usnea. The life cycle involves both vegetative as well as sexual methodologies, methods, but the vegetative reproduction is the far more successful and common one. And as far as this vegetative reproduction is concerned, this method ensures the direct propagation of the established fungal-algal partnership. So it is the main mode is the fragmentation. This is the main mode of reproduction. You have seen the long hanging thalli of Usnea, which are brittle when dry. They easily break into small fragments or small pieces due to mechanical forces like strong wind, heavy rain, or sometimes through foraging animals. And the process, when you consider each fragment containing both your algal as well as the fungal cells, so that they are dispersed by wind to germinate on a suitable substratum where they absorb moisture, because there is a fungal hyphae. So on landing onto a suitable substratum or substrate, like the bark of a tree in a very humid area, it can absorb moisture and it can attach itself, start growing into a genetically identical new Usnea lichen. And we have to be very clear with this, because it is vegetative reproduction, and so the parent genetical material is passed on to the offspring. So the new Usnea lichen will have the exact genetical characteristics of its parent plant. And now we pass on to the sexual reproduction part. Only from the microbiont you can see the sexual reproduction. Kindly recall or recollect, a lichen is a composite organism having both fungal as well as algal partner. And the algal partner is referred as a phycobiont or photobiont, and the fungal partner is referred as mycobiont. Please note, the sexual reproduction in lichen is happening only in the mycobiont partner, not in the algal partner. So how it takes place? First of all, there will be a formation of fruiting bodies, followed by spore production. Once the spores are produced, they are dispersed, and on falling onto the suitable substratum, they will germinate, and there will be a major challenge for this fungal partner, which is being reproduced through sexual means, that is the microbiont. It has to find the right algal partner to make a new lichen thallus. This is a very challenging part. So here the sexual reproduction, it's more complex, and it is uncertain process that only involves the fungal partner, and this is taking place with the help of the structures that you see here, which are referred as apothecium. Singular apothecium, plural apothecia. A closer view of apothecium, and yet another closer view of apothecium. So a sort of plate-like or a sort of saucer-like or cup-like structures, not a deep cup, it's a structure. And let's see the structure of this. Here you can see the line diagram of apothecia, and this saucer-like structure, it has got a long stalk, you can see it here, and you have certain topmost layer which we referred as hymenium here, and in hymenium you have two structures. One is asci, singular ascus, and these are the ascus. Just a second, I will take a color pen. This particular layer, I will take another color. This particular layer, the hymenium layer, and you have many bag-like structures, and these bag-like structures, a single structure is ascus, and there are many, so we call it as asci. And in between these two asci, you can see some linear structures. These are the ones. They are referred as paraphyses. And what is the function of this asci? They produce ascospores. And now detailed structure you can see here. These asci, they are produced by ascogenous hyphae. And now kindly recall that ascogenous hyphae or hyphae which are having two nuclei, one from the male strain, another one from the female strain, formed because of a phenomenon namely plasmogamy. Here there is no karyogamy, only the plasmogamy. That means you have the two nuclei lying side by side, but yet there is no fusion. So this kind of a hyphae holding two nuclei, referred as dikaryotic hyphae, formed after plasmogamy, they are the reasons to produce these asci. And now in this particular ascus, karyogamy is taking place. That means you can see here karyogamy is taking place. That means these two nuclei which were lying close to each other without fusion is now getting fused inside this ascus or asci. That we call it as karyogamy, that is fusion of these two nuclei. Karyo refers to nucleus and gamy, karyogamy, and then it forms zygote. Once the zygote is formed, it undergoes meiotic division. That means from this one diploid nucleus you will see 4 haploid nuclei. After this there will be mitotic division, and then these 4 becomes 8 nuclei that you see here. So you can see, I will take another color so that we will be able to see. See here 1, 2, 3, 4, 5, 6, 7, 8 spores. So these are the ascospores. So that is how the structure of apothecia is discussed, and the layer which you see, a fertile layer, that we refer it as that. Okay, so this fertile layer where you can see the asci or ascus, that is referred as hymenium, and you have a margin for the whole of this apothecium by the fungal hyphae for protection as well as for absorption of moisture and other things. And you have beneath this hymenium, hypothecia will be present, where you can see these asci or paraphyses used to present. And then as usual you have medulla here in cortical region, where you can see loosely arranged algal, fungal hyphae. And then when you take a single ascus, you can see this whole structure is referred as an ascus, and you have paraphyses here, and then you have ascospores inside. So all these are ascospores. You will be having 8 ascospores here, 3, 4, 5, 6, 7, 8 ascospores. So this is how the sexual reproduction is taking place here. So once they fall on a particular proper substratum, they will produce the new lichen, provided they have to have a compatible algal partner. So when you see the mind map of Usnea, you have the common name for this Usnea, old man's beard or beard lichen, fruticose lichen present on the tree bark. So we can call it as a corticolous lichen. It has internal structure, it has a central core followed by medulla, and then you have algal layer, otherwise referred as gonidial layer, and final outermost protecting covering, that is your cortex. And then there is a vegetative reproduction, far more successful, primary mode, and simply through fragmentation and sometimes through soridia and isidia. And then sexual reproduction is through the production of apothecium, which is a fruiting body where you have asci or ascus, which are referred as spore sacs, and ascospores are produced. So there will be again these ascospores finding on a new substratum with a right fungal partner. This fungal algal partnership will be established. There will be mutualism, and we know that it is a pioneer species, and they are also the bioindicators. So this is the simple mind map of Usnea. Take a notebook, have a pen with you, listen to this lecture 2-3 times, and whatever the important terminologies are there, kindly make a note of it, and this will be easy. I made it very simple so that you can learn it properly. Please watch and take mind maps so that the subject stays in your mind. Thank you once again.